So these are the notes for our foldable cell cycle. And we begin with our two folded pages, and yours may already be glued, but if not, you're going to overlap one corner, and you want to be very careful that you don't overlap the fold, so it should still be able to fold once it's glued down. Making sure it can still fold once it's glued. So the whole thing will fold all together, and we're going to begin with the inner part folded up and then the outer part folded up. You're going to need some markers for this. You're going to need 4 different color markers and your regular writing utensil. And it doesn't matter, pick one of the colors. You're going to write the title. These are cell cycle notes.
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material.
And then we're going to put a note. This is going to help us remember cells come from other living cells. Open the whole thing up, and here's that other one folded. We're going to leave that one folded up for right now, and we're going to start in this top corner, and we're actually going to draw a pie chart, and it's going to show us the amount of time that the cell spends in each of the stages of the cell cycle. So it doesn't spend equal amounts of time. So each of these squares does not represent equal time. In fact, one of these squares gets most of the time.
And what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and it's replicating its DNA and carrying out some of its cell processes. That's what it spends most of its time doing.
We'll draw a big circle for our pie chart. And I'm going to draw it like it's a clock at 2 o'clock. And then this smaller pie slice, I'm going to split a little bit less than 1/4. The majority of the time is this first part, which is the stage called interphase. It doesn't matter what colors you're using here. Of the 2 slices left, the larger of the 2 is mitosis. And the smallest is the longest name, Cytokinesis. We're going to label the 3 stages here. And you're going to pick your color that you're going to use as our label and hint color. So I chose my blue. So I'm going to label interphase. Not too big because we're going to have to write some things underneath it. And you notice the squares are kind of divided into triangles. So there's interphase with a little number 1 next to it. Then down here, and this is the one that would fold open, we want it so there's 2 sides tucked to it. This is going to become mitosis, number 2. And then number 3 Cytokinesis. I'm going to put that one at the bottom. We're going to zoom in on Interphase first, and you can leave yours folded flat. I just have to fold it for the video. But we'll zoom in here. And we're going to draw a cell in Interphase. I'm going to use my regular writing utensil. I'm going to draw my cell in this bottom part of the corner, bottom part of the square. Nice big cell. And it's got a very important organelle in here. Very large organelle. So what organelle do you think that's going to be? Well, that organelle, nice large organelle, is actually the nucleus. And you should have 3 other colors left, so we only used 1 color so far for the title. So you're going to take 1 color, and we're going to make an organelle out here that's important for cell division. The centriole. So if it has centrioles, what kind of cell is it? Animal or plant? If you remember, centriole helps the animal cell divide. It's a good job. Then my remaining 2 colors are going to be something that's inside the nucleus. So the nucleus has several parts. Nuclear membrane, which we drew. The nucleolus, so nucleolus, if that's how you remember it. But we'll learn more about that later on in the year. So this is the last part of the nucleus, the chromatin. So you're going to use 2 colors to represent chromatin. And it's just kind of squiggly lines right now. So I'm going to label that my chromatin. My cell here, going through Interphase, is doing several things, important things. It's going to be growing. I'm using my regular writing utensil to list these. It's going to have cell growth. It's going to replicate DNA, which means to make a copy. And it's going to generally get ready to divide. Prepare to divide. Which means it's making copies of everything else, like mitochondria and ribosomes and all that stuff. So there's enough to go around. So enough for 2 cells to be made from it. Now I actually have some phrases to help us remember these stages. So interphase is the in-between stage. And I'm going to take my title color and draw a little arrow. So I'm crossing over with my arrow. You see we go from interphase to mitosis. This is when the nucleus divides. So we call it nuclear division. To open up and look at mitosis. So opening wide up, we're going to start at that top corner. And remember, you don't have to fold yours. I just fold it for my video. And this arrow that we had started up here and got cut off when we opened it up, I'm going to actually finish the arrow right here into our first step of mitosis, which is prophase. Using my title color, and my hint for prophase is partners. So let's draw our cell again. Use your regular writing utensil. The bottom corners looks like a good spot for it. So our nuclear membrane, and this one is actually starting to disappear. And by the end of prophase, it'll be gone. Label that. The centrioles are starting to head to opposite sides of the cell, but they're staying connected because they're partners. They stay connected with each other. And these things that they connect with are these spindle fibers. Inside of the nucleus, the chromatin condenses and it forms something called a chromosome. You've learned about those before. So condensing is just a fancy way to kind of say folding.
Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope: chromosomes. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled into chromosomes.
And I had my 2 different colors, and notice I have 2 of each color, 2 chromosomes of each color, because they are partners. Actually pause here and take a closer look at those chromosomes. So I'm going to fold this back up. You can see mitosis there. So we're going to fold that to the side and we're going to draw a chromosome. So a chromosome has 2 sides that are held together by a piece in the middle. And do you remember what a chromosome is made of? It's made of DNA, so I'm going to label that. And you can use the markers, doesn't matter what color. Then the parts, each side is called a chromatid. And each chromatid is an exact copy of the other one. So when the DNA is replicated, when a copy is made, it's held together. The 2 copies are held side by side until it's ready to split. And then the middle piece that holds the 2 chromatids together is the centromere. So that's a little closer look at what a chromosome is. So now we can open back up our mitosis. And we've done prophase, the partner stage. So we got partner chromosomes and we got partner centrioles staying connected by spindle fibers. Here's a picture with a few cells pointed out going through prophase at different points. So one has the nuclear membrane gone. In this picture, can you find the one going through prophase? If you guess this guy in the middle, you'd be correct. Now, we go to metaphase. And the hint is middle. Looking at this picture, can you guess why? So, draw my cell. And the reason the hint is middle is because the chromosomes are lining up right in, probably guessed it, the middle. Meanwhile, the centrioles have moved to opposite ends of the cell, and we call those the poles. Like the poles, north and south pole of a globe. But they are connected to the middle, the centromere, of the chromosomes by the spindle fibers. So we have chromosomes lining up. In the middle, and spindle fibers connect to centromeres, which are the middle of the chromosome. This picture is a computer rendering of chromosomes lined up in the middle, and they're so long you can see they're overlapping. That's the blue in the middle. The green is the spindle fibers. And this is just a cell under a regular light microscope, and we can see the chromosomes lined in the middle. Alright, so moving on along, now we're bottom right corner of the mitosis pull out. Have our arrow coming down into here. And this one is Anaphase. And our hint is apart. So the cell actually stretches out. So I've got a nice long cell. Centrioles are still on the opposite ends of each other, but they start to reel in those spindle fibers that were connected to the centromeres. And when they do, they pull the chromatids apart from each other, and they get kind of dragged along, and they look like a banner or streamer being dragged behind someone. So those centromeres split apart when the spindle fibers pull or get pulled by the centriole. So the chromatids are pulled apart to opposite ends, the opposite poles. And then we notice the cell is stretching out. Here is those green spindle fibers pulling the blue chromatids to opposite poles in Anaphase, and once again under the regular light microscope. Alright, then we go on to our last part of mitosis, Telophase. And my hint is 2 nuclei, because that's what we end up with at the end of telophase. So telophase actually overlaps with the next part of the cell cycle, cytokinesis. So during telophase, the cell starts to pinch inward. So the cell membrane starts to pinch inside. Kind of looks like a peanut there. And our cell starts to develop nuclear membranes again, so they reappear. Inside the nucleus, my chromatids are going to stretch out. So we call that relaxing. So they unfold a little bit and they go back to the chromatin state. So they're stretching out. And the centrioles have done their jobs. They're just kind of hanging out on opposite sides of the cell. So now we're ready to head to cytokinesis. So I'm going to close back up my mitosis. And see that arrow? And kind of extend it from the bottom here. Cytokinesis, my hint is cells, plural, because at the end we're going to have more cells. So I'm going to draw my two cells. They're not as big as the original cell was. But there are two of them. Two nuclei in there. And by the end of cytokinesis, each daughter cell is going to get half of the organelles. And a full copy of DNA, because that's the instructions. So cytokinesis is when the cytoplasm divides, the rest of the cell divides. So I'll draw a centriole in each just to show how the organelles in the cytoplasm get split in half. And the chromatin in the nucleus is back to our kind of squiggly line state. You can't really tell much about it. And we have two identical daughter cells. So let's fold this back closed. So I got my mitosis down. I fold the edges together. And we had cell theory on one. I'm going to flip up the other way. And I'm going to put my little memory trick. I just remember a funny sounding sentence. I P mat C. So not I C mat P, but I P mat C. And P mat is mitosis. And that's how we go from one cell to two identical daughter cells.